तुमसे अभी यहाँ से जाता ये करते रहोगे
जी,
तुम्हें फिर अभी इधर से
हाँ, हाँ।
मूवमेंट करते रहोगे
हाँ।
कितना कितना बार करोगे?
एक या दो दिन का ज़्यादा।
Hello, I am audible, right? Okay, so we'll wait for one or two more minutes and then start. Okay.
बातम भैया, हम कोई सा भी रनिंग बफर यूज़ कर सकते हैं ना? रनिंग बफर कोई सा भी यूज़ कर सकता हूँ, 1x यूज़ करना पड़ेगा। हाँ, ज़्यादा पुराना नहीं होना चाहिए, नहीं तो तुम्हारे जेल में उनके भी बैंड्स आएंगे प्रोटीन के, क्योंकि उसमें ऑलरेडी डायल आता है पहले से। ओके, अगर तुम जस्ट अपना टेस्टिंग के लिए कर रहे हो और ये मतलब तुम्हारा एक्चुअल में आगे नहीं जा रहा, देन यू कैन यूज। ओके, ठीक है।
Okay, so let's start. So today is the week 5 for the introduction to proteogenomics course live session. So let me share my screen and just let me know if you can see it. So the screen is visible, right? Okay. So yeah. So already we are looking at the different case studies for the proteogenomics approach, that how the different techniques have been used and how the data and the data are analyzed to find out that how both the techniques are integrated to understand novel findings for a particular disease. So today I thought to just give this paper, about this paper which has used the proteogenomic approach to understand the breast cancer tumorigenesis and to understand and find some novel targets. So you can see, so it can be seen that they have performed acetylation phosphorylation, that is the post-translational modification of the proteomics, as well as the whole exome sequence RNA-seq and the proteomics, and they could classify, then identify some therapeutic targets and all. So what is this classification? The classification, and for this week, if you all have gone through the different the videos of this week, so you know that the classification as well as the clustering plays a major important role. So just can you say the difference between clustering and classification? So what, when do you cluster, and how can you classify? Anything would be? No, then okay. So let us think like when you classify and when you cluster. So if there are two different baskets and you have fruits and vegetables, then you want to classify, right? That you this is a fruit and this is a vegetable. But if in among the fruits or the vegetables you want to differentiate, then you will try to find the closeness and then you will cluster. So that can be you, that is a very broad example or vague example. So you can cluster. So basically one is to using a supervised method you will try to classify that this belongs to this particular class, and another is that basis on the closeness or how much it is related you would like to cluster or group them, that these are the basis of the similarity features that you would like to cluster them. So now here in this paper and this study you can see that different clustering techniques have been used on the basis of the PAM50 and the NMF. So you can see that their clusters have been formed. So on the basis of these genes, so you can see that basal, HER2, LUME, LUMB, luminal A, luminal B. So on the basis of all those, their different clusters have been performed. So basis of the closeness on the intensity level or the expression level of those genes or those proteins, it has been clustered. That this is a similar. So these are this forms this group. On the basis of it, it has formed. So you all can see that this is how the cancer of a same type like the breast cancer could be clustered or could be differentiated into the different subtypes based on their specific gene expression markers or the protein markers. Another thing is that you can see that on the basis of the PTMs, their protein expression and the acetylation, again these clusters and what are the relative expression values. So you can see that there are some of the samples which can behave differently, like the uh, some portion of the luminal B1 can behave like luminal A1, and hence you can see there are patches of the there is an overlapping. So you can go and look at it that what why those samples behave like that way, or why they are closely related to, they are classified into a particular group but has been used or has been looked at a and is with on the basis of the expression of the markers, it is looked at a different group. So for this the clustering becomes important to understand, and that the protein or the sample that we are looking it as a particular marker, whether there is any interesting they are showing any interesting behavior or not, or are they grouping according to the classification only, or they are changing. So this you can see that basis of the pathways, different pathways and their intensity and the proteins related to this intensity and how they are clustered among the groups. So this is for the clustering. So the next thing is that we had started using metaboanalyst, if you all remember in the previous week. So here we will try to look that how we can with our own data set, how can we form cluster, or either we can make this kind of graphs or not, whether we can assess or not. Okay, so we will now with the no, the clustering is dependent on biomarkers. If you the thing is that if you will get a proteome or genomic sequence profile, okay, of if let us take that there are two groups, one is control and another is diseases. So controls whole proteome profile or the RNA sequence profile. Similarly, you will get the protein profile or the proteome profile of the uh, disease patients. Now we will look for the in the cohort that how much is the similarity. So you will try to cluster on the basis of that. That okay. So this control samples is this has this expression. One sample has this kind of expression values. So now again you will see that okay, another group of sample has similar kind of expression. So you try to group those. Similarly, if another sample is has somewhat different kinds of values, so you will try to group it differently. So in this way, you will try to cluster those samples. So the clusters can be of like hierarchical clustering or the K-means clustering basis of that. So there are different kinds of clustering techniques, but generally it tries to group those accordingly. So are you clear about it? So biomarkers is that is those things that is differentiating. So those set of proteins or the genes that will differentiate it. Okay, so you have to find out those. You have to be at the go to is to find out those genes that are behaving differently. So on the basis of those, ultimately what we look at, the majority of the proteins or the genes will remain same across the board, and if you see that there will be no difference, but there will be certain set of genes or the proteins that are behaving differently, and hence why their two groups will form or the cluster will form. So on the basis of that we can identify those markers. It is basis on the gene expression values. The thing is that on the diseases like cancer, it can cause due to overexpression of certain genes, due to mutation of certain genes, due to differential, that at the gene level it is okay, but the protein product is not formed at the sorry, what you say, that at the genomic level. So that's all thing can happen. So means at the RNA transcript level there is something has happened, that protein is not forming. So hence all these kind of things can happen. So it is on the basis of the gene expression or the protein expression or their modifications or the mutations, the different generally we take the expression values, all those effects finally in their expression values. So on the basis of those, we can identify those biomarkers. Okay, so now uh, I data set. Okay, sharing the data set. So let us do it. So first thing already you know. So first you will tell me what steps to be followed, and then we will start looking at that if we can cluster and all or not. So analyst. So let me know till this page. Okay, and what to do after that. You have got the data set, right? Okay, so have you reached till this page? Why? But it shouldn't be an error. Okay, you have the previous week's data set. If you have the previous week's data set, then also it will be okay. You have the previous week's data set. Okay, so upload that only. It's not an issue. So once you have, just let me know. Done. Data set is showing error. One second. Now check, because I uploaded, it's not showing any error. Yeah, you can share. Are you sharing it? You go to MetaAnalyst once, type in the Google. Are you typing? Okay. And let me do it once again. So this is,
ऐसे कैसे नहीं खुलेगा? कोई नहीं, मुझे नहीं पता है, अभी रिसेंटली ये बंद हुआ होगा।
this is where you go.
मैं वो बड़ा वाला फीडबैक का है, वो जिंदगी में बंद नहीं करने का, और मतलब देख के बंद करने का, कुछ है चल रहा है या नहीं चल रहा है।
So you have done till here, right? Okay, so now it's fine, right? Okay. So after this, what steps should be done now? So there is written that a number of missing values have been detected. So what needs to be done here? You remember, or Okay, so here we need to impute, right? So first we will go to missing values. So how many? So which have greater than 30% of missing values? We will remove those and we will estimate these values using KNN and feature wise. Done. Go to the missing value, impute the value. So why we did wrote 30%, and the imputation is done group wise or sample wise? Yeah, group wise or sample wise. So the imputation is always done so the these groups and two groups. Okay, so then proceed. So now what we need to do is that now, hello. Yeah, so median is one kind of normalization, so that the sample means come to a particular place, and after that what happens is that we can uh, find out the differences, that which features are differentiating, are there any significant features or not that are differentiating the two groups, right? So transformation is generally for our biological data we perform a log transformation so that the scales or the intensity levels that are different, that comes to a scale, right? So after we will normalize it, and then we will do a visualization, so we can see that it is fairly normalized. So before normalization and after normalization. Then after that we will proceed. Okay, so now there are various fold change analysis, t-test, volcano plot. So first we will go for the correlation heat maps. So now can you tell me what does it mean, this heat maps? Have you gone to this correlation map? Yeah. Done na till that. Tab is stuck. You just click on go to another tab, and but that got uploaded, right? Okay, so let us then you can check it otherwise, so you can just have a look at it now. Okay. This is a heat map, this is a correlation map, right? So what you can see is that a bit is a correlation between a disease 2 is 7 for any kind of biological samples. So what happens is that so the two type same type of sample will have a highest correlation, but there are different groups like control and disease will have slight difference because that particular features are differentiating those two samples, and that's why they are in the two different states. So this is the basically the difference. So we first check the so after that what we will check is the PCA plots. Anyone? So if there are you have two groups of samples and you have run in two different batches, so how you can determine that there is a batch effect or not? One way of determining the batch effect or not. So you will definitely do PCA, right? So PCA will just cluster in an unsupervised way, and then you will look. No, PCA is always unsupervised clustering. Okay, so it is not supervised. So it just on the basis of the features, basis of their its components, it will cluster it will give you the clusters. So now after that you will go and look at it, that okay, so your two cohorts like control and disease are different or not. So if you have done in two batches, are they all mixed up together or they are apart? So if they are too distant, then you will say that okay, so there is a batch effect exists and you need a batch correction and all. If they are mixed, then it's okay. So if you have two groups, so it should be clustered apparently, but always it is not the case. So you need to perform other kinds of analysis. uh, for the real data it becomes difficult for getting two different cohorts or clusters almost differently. Okay, this is a PCA plot. Okay, so now but we will today go. So I think you have the previous week you had we had looked at the volcano plot, remember? So volcano plot consists of y volcano plot's x-axis contains what and y-axis contains what? So volcano plot checks on the significance, that is the t-test or the p-values, as well as the fold change. So x-axis will contain the log to fold change and the so now we can look at it. We all can play. Now we will go to the hierarchical clustering. Yeah, so you were giving the data source as the normalized data. Okay, so you were auto scaling the features, distance metrics. There are different kinds of distance measure, so you will find out the distance. So it is an hierarchical clustering, so you can read about it. So what it will do is, so it will measure the distance based on the features and then it will try to cluster. So clustering method is what? So control, so you can do not cluster samples, or you can allow to cluster the samples, and you can check on it. Similarly, for this protein, you can see that it is upregulated in the disease class and downregulated in the normal class, right? So you could see the heat map, so how it has been clustered, and you can see that basis on the distance it has clustered the both the sample disease cohort, and you can identify these two differences, right? So this is, you can find, right? So now if we go for k-means, so you can specify your cluster number. See, so it is clustering according to it. This is one cluster, this is the another cluster, but you can see that it is not getting divided according to what we want, right? It could not do it. So hierarchical clustering, you could see that it is giving a very, on the basis of the distance, it could distinctly cluster it and also find out the features that are getting differentially regulated. But there is a it is showing this uh, different types of clusters. So you can try out different. Yeah, so the dendrogram also you can look at it. So here it was also measured, so you can see that one disease is behaving as a control. Okay, so in this cohort it has differentiated both, but one disease you can see that it is behaving like a control. So now this becomes interesting to look at it, that okay, so do you want so why it is behaving as a control, or what could be the reason? So different algorithms, different techniques, you can analyze your data and can try to find out that if both are getting differentiated, or uh, if there some particular this one which is behaving differently. Okay, so you can see, so here also you can see, so if you can see that whatever paper what we showed, that the different kinds of things from here you can see. So these are the diseases, and this is the control sets, uh, which have been which are getting the clustered, and what are the so this is just a sample wise. Let us look at the feature wise. So this is according to the features. Okay, so the number of proteins are there. So what is, how they are behaving in the different samples. Okay, this is a protein wise correlation. So the protein, so how one protein is for at the particular sample and at a different sample. So this is the for the feature wise correlation you can look, but generally the correlation are looked at the sample wise to look at it, that how their correlation, generally the biological data gives a higher amount of correlation, and from here you can make the feature wise dendrogram, as well as you can make the heat maps. So any question till here, anyone? So the data set I've shared the data set. From here you can try to build it. So if you look at, if you do the do not cluster here, so then also you can find out the differences of the proteins and the difference in the different cohorts. Okay, so if you want and find out the differentially regulated proteins, so you can see that these are the number of significantly upregulated proteins, and these are the significantly downregulated proteins. And from here you can get on that which are the proteins, what is their fold change, what is their log to fold change, what is their raw p value, what is their minus log 10 p value, and finally you can uh, find out that what proteins are there, and you can do a biological analysis. Okay, fine. So this is one of the tool metaboanalysis from where you can do the different kinds of analysis. So similarly, I would like to show another software uh, that is also used heavily to analyze the proteomics data, is Perseus. So I'll give you the link. This is, you have to download and install, and then you can uh, do it. So you all can go and download it and can use. I'll just show you that how this same can be used here also. So finally, what you can do is that take the data, so it generally, if the names are same, it takes one. So let us just I'll just give you a basic demo that is mostly the same thing, just you can use different things, use different software. So if you can go, so if you go for the visualization, so you can see that there are two, it is not normalized, data is not normalized, one is showing control, another is disease with difference. So if you just go normalization, and same if you do a median sample wise normalization, and then if you go and do a visualization, so now you can see that two medians are same. Okay, so here it is, the data is not normalized, that is the missing value needs to be imputed. Here also you will measure that the minimum 70% values, if it is there, then only it will be there, otherwise it will reduce it. And then after this you will do is that you will impute it. Okay, so I would like to show the, here also you can perform hierarchical clustering. Let us look at the PCA best. So you can see, okay, so the difference in the component 1 and the component 2, So this is another software from which is used generally to perform the same things what has been done in the meta-analyst. So you all can try out this also and use the same data set and can give different names and can try out the different normalization techniques and can visualize and perform the clustering and the different parameters. Then you will understand that how your data you can analyze and visualize and can make out the biological sense out of it. Okay, so I have given the link here, so you all can just with your academic license you can download and can install, and it's very easy to use and also user friendly. Okay, so any question? So one is Metabo Analyst, which is very friendly and easily available in the web based tool, and another here you can use it. No question, right? Just based on it. Okay, so so as we are discussing every day, so about the tools, I don't know if you all are looking at or not. So for the genomics data analysis, so we need a certain kind of steps to form the database. So if you are having, if you want to analyze your proteomics data set, so generally it's in the approach we need a database, and if you need a patient specific database, so you need a customized database. So what is the main principle or goal behind this proteogenomics approach? So here if you get the databases, so you can translate it and perform the proteomics data analysis. But since you want to have a specific mutated or the specific mutations, if you want, or the proteins you want to capture it, so you need to analyze the genomics data, identify the variants, and then prepare a customized database. So till previous week I told that you will first check the quality of the data, then you will align it with a reference genome that can be your normal genome from your normal region as well as the genome from your the whatever is present like HG38, HG19, etc., etc. So now after you get the variants, now you need to annotate those variants, and for annotating the tool and search is the tool is the ANOVAR. Okay, ANOVAR is one of the kind of tools. There are many tools present, but ANOVAR is one of the most widely recognized tool. So it will annotate your variants. So what do you mean by annotate your variants? Like another SNPF is also there. So what do we mean by annotate? That is it a missense mutation? It is a deleterious. It is a non-synonymous mutation. It is a synonymous mutation. So it will let you annotate the variants, and after which you can prepare the customized database. So ANOVAR also is helps in you in understanding the annotating those variants that you get after the aligning. So after visualization of the alignment that chromosome wise, so you will try to annotate those variants according to the different categories, and after annotating or after annotation only you will try to go and prepare the customized database. So okay, so are anything you all want to ask or anything? Is there any questions? Okay, so everything is clear till now. So you have understood that what is this clustering technique and why it is used. So basically you need to differentiate between the two groups, control and disease, or the treated or control or anything, and identify the differential biomarkers. For instance, if you get a customized database and identify those proteins or those mutated proteins, and so then you will need to identify that, then you also need to cluster this. If you get the proteins or the mutations, like this is the mutation profiles of 10 patients and these are the mutation profiles of the means these are the profiles of the 10 control samples or the healthy individuals, then how the mutation profile would cluster your samples. So that's why the comes the principle of clustering and identifying. And if you are not able to cluster it or group it, then you cannot identify that what are the specific markers that are driving the disease, or you cannot make the biological sense out of it. Another thing is that rather than clustering is that once you can also have a supervised way of analysis, where you can say that these are the amount of samples that you need that are belongs to a particular group, and you can classify that, okay, so this with this set of markers, with a set of a panel of biomarkers, can I classify that this patient will be a control one or the disease one. Similarly, this for instance, now during the COVID-19, it was very much important to understand that a patient will become severe or not. So if you can just remember, so there was a need to identify this sort of biomarkers that can tell that whether a patient will become severe or not. So if you can get a certain sort of biomarkers that will let you know that a patient will become severe, then you can take extra precaution and all. So that's kind of biomarker. These are the principles, and these are all the needs of the identifying these different biomarkers and classifying and grouping the different cohorts, so that we can identify and understand the principles behind for a particular disease. So if you all have question, then we can close this session for this week. So do you have any doubt or any question? So you all are comfortable with meta-analysis, right? So would we can just go ahead back and look at it, why, what was the error, because it is the same data set that I used. Okay. So just, if it still persists, then we can look at it in the next week again. Okay, or you can write in the forum, or I think you can write also if you find anything that it's still existing, or otherwise you can try on your own once. And also you all can try Perseus also once. Just it is also very easy to install and all. And have you all installed Protege and used it? In the lecture you all found out na Protege, you have tried to install or installed it? Okay, so how, so you found it convenient, right? Okay, so you can try out this different software also. So one you can try out in analyzing meta-analysis, one you can try analyzing in Perseus, one you can try out analyzing in Protege, and you can see that the different parameters, the different algorithms, each tool possesses, each tool has a own set of algorithms, different algorithms. All things are not present in one tool, so you all can become comfortable also, and also you can try out different types of analysis and can look at how data is changing or how data is behaving, right? So you all can do that also. So you can try Protege also. If you face any difficulty, we can discuss in the next week also. Yes, so yeah, so you can try out these different methods. So yes, so okay, thank you. So thank you everyone for joining. So we will close this today's session for the week 5, and we'll again meet next week on the week 6. So I'm closing the session.